Chapter 6, Molecular Information, Flow, and Protein Processing. Section 6.1, DNA and Genetic Information Flow. Now we need to keep in mind, okay, a lot of this, what we're going to be talking about here in the first few sections is review information from intro cell and molex, cell biology, genetics. So bear with me, okay? May have been a while since somebody's had one of those courses. So we need to go over this information, make sure everybody's on the same page. Keep in mind that when it comes to genetics, everything is based on the gene. Okay, the gene is the blueprint on how to make something. Okay, we're going to see that the genes aren't alone. They come as what they refer to as genetic elements. It could be large or small things that we refer to as chromosomes. Okay, a collection of genes. Since not all organisms have a singular chromosome, we discuss things in terms of the genome. All chromosomes, whether it's in a nucleus, a nucleolus, the nucleoid, or an organelle like the mitochondria, the chloroplast. DNA is going to be the blueprint for all living cells. Yes, I know, chapter 5, we talked about viruses. Viruses don't always have to use DNA. Certain viruses are going to be RNA-based, but for all living cells, prokaryote, eukaryote, DNA is king. RNA, this is where transcription and translation events come into play. Central dogma, as you see there on the far left. DNA replicates DNA. DNA acts as a template for replication, the making of new DNA. Transcription means that you are transcribing. You are taking the DNA and you are making a RNA copy. Okay, notice it doesn't say messenger RNA. It could be messenger RNA, tRNA, ribosomal RNA, small RNAs, which talk about chapter 7, chapter 8. The messenger RNA is going to be read by ribosomes for translation. Read the information in the messenger RNA and link together the amino acids. That's translation, the making of the polypeptide. Remember, the nucleotide, you have the nucleoside, which is nothing more than the sugar, and the base in the nucleotide, which is the phosphate attached to the 5' carbon, the sugar, and the base. We don't show the bases here because it's showing this generic, because there are 5 different ones. It will always be a pyrimidine to a purine. 1 cyclic ring, 2 cyclic rings. The nucleotides come in 2 flavors, DNA, RNA, ribonucleic acid, deoxyribonucleic acid. The sugar, the nitrogenous base, the phosphate. You have to have all 3 for the nucleotide, but some metabolic functions just require the nucleoside, the sugar, and the nitrogenous base, the purine or the pyrimidine rings. You don't need the phosphate groups. A, G, C, T, and U. Adenine, guanine, cytosine, thymine, uridine. A, G, C, T are DNA. A, G, C, U, RNA. For you to know if you're looking at a question, homework question, test question, is what I am dealing with. This sequence of nucleotides. Is it DNA or is it RNA? Look for T's, look for U's. If it's AG, TT, AG, CCC, well, there's T's there. means it's DNA. If it's A, G, C, U, 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 G, C, C, G, G, whatever, RNA. You've heard since you were a little kid, the double helix. Watson and Crick, the double helix. All right? The double helix is the strands of DNA nucleotides. Each nucleotide held together by a phosphodiester bond. The first nucleotide, via its 3' carbon, is attached to the 5' carbon of the next through that phosphate group. So it goes sugar, phosphate, sugar. The structure here, the primary structure, is just the sequence, A, G, C, T's, and U. J, G, C, T's, or A, G, C, and U's, if you're talking RNA. They're complementary. A only goes to T, G only goes to C. Five-sided sugar. There's the 5' carbon. 1, 2, 3, 4. 5 is up there. 5 has the phosphate group. The 3' right there, 3', remember I said it's connected to the 5' carbon of the next sugar via this phosphate bond, this phosphate group. Sugar, phosphate, sugar, phosphate, sugar, phosphate. 2, make it so that we know as we are talking about DNA sequences. So we have our orientations correct. You will see that many textbooks, all primary literature articles, if they are giving a sequence of nucleotides, will say 5', 3'. It'll say 5', A, G, T, C, 3'. So that this way you know this is the orientation they're talking about, not the opposite. So this way you know that this is the first nucleotide in the sequence. Because if I just gave you a sequence saying CTGA, is it CTGA up here, CTGA, or is it CTGA going from the opposite end? And we always start with a 5' end as we are listing them out. 5', G-A-C-T, 3'. Now, this double helix, as is shown on the last figure, is held together by hydrogen bonds. Hydrogen bonds being some of the weakest bonds we have. But they're cumulative. One hydrogen bond, easy to make, easy to break. Billions of hydrogen bonds? Well, now the double helix seems to have some staying power. Since they're complementary, remember A goes to T, G goes to C. So if I just tell you one of the strands of the double helix, logically you can figure out what the other one is. I tell you 5', 3', 5', GCCAT, you know 3', you know the other side goes, you know, G, G, A, T, I forget what the other was. Since they're anti-parallel, 5', 3', pointing this way, 5', 3', pointing that way. A to T, G to C. A lot of times students seem to think that the double helix, you just take that ladder-like that's drawn on the last slide, and you just twist it, and that's how it looks. No, you take that ladder, and then you wrap it around a pole. That's the double helix. Because of this double helix, you end up with what are referred to as major and minor grooves. Major grooves, this is where proteins that interact with the double helix can come in and interact directly with the AGCT nitrogenous bases. Now, this is not set in stone, okay? These major and minor grooves, the helix twists, so that way, this this AT base pair right there, right now is in a major groove, gives another twist because the proteins around it shift. That twist changes, alters a little bit. What was in the major groove is now going to be part of a minor groove as more and different nucleotides are exposed to a major groove. This is not a static shape. When we look at the size of the DNA, have to keep in mind one base pair, A to T, G to C. Take 1,000 of those, you now have one kilobase pair, or one KBP. Take 1,000 of those KBPs, 1,000 times 1,000, you now have 1,000,000. Or what they refer to as one mega base pair, one capital M BP. KB, MB, and then when you get into eukaryotes, you start measuring, counting things in billions with a B. E. coli's genome, 4.64 mega base pairs, 4.64 million base pairs. That's a lot, even though it's a lot there. What we see to make DNA fit into such small cells is they're going to do what's referred to as supercoiling. Yes, you have the double helix, but then you're going to start twisting the helix around so that it can fit more into a smaller area. Think about it. You've always seen it drawn like a circle. Okay, you remember playing with rubber bands or that string, that cat's cradle game you played as a kid? Well, that's what cells do, especially bacterial cells do with this ring of a chromosome. They'll take it and they'll start folding it back in on itself, forming these circles back in on itself. Enzymes will come and cut, repaste. So now instead of coming up and around over, and then under, comes up, goes down, around, under, up, and over, and it just twists, and it keeps twisting and twisting, and it'll take this and it'll twist it again, and again, and again, and again. This is what supercoiling is. You take this big circle and you just start twisting it in on itself, knotting it up in specific ways, bracing it with proteins. All of this is still accessible. Just as this is accessible to all the polymerases and whatnot, DNA polymerases, RNA polymerases, so is this. This is big and a pain in the ass to keep track of. This, everything's been folded. This will take up less space than this does. This, because of all the supercoiling, is actually more stable than this is. Now, because of the way the artist drew this, this looks like it's so much bigger than that. No, they just drew it bigger because if he drew it to scale, the actual supercoiled thing would, look like this. And, well, this looks more dramatic than that. How's this work? How's all this supercoiling work? Well, first, you're going to have a set of proteins. Proteins that you've heard the names before, topoisomerase, DNA gyrase. Topoisomerase, you've heard it in other courses. This is what, removes, the knots, this overcoiling when you have replication occurring. Well, yeah, it removes them, but it also puts some of those major coils in the way to help package things down. Depending upon which way things twist, it's either referred to as negative or positive supercoiling. Under normal conditions, the DNA double helix has a right-hand twist. Okay, if you go back and look at the figure in the textbook, it goes up and around. They call it right-hand twist because it goes this way. So the left-hand twist, this is my left hand, it would be coiling the other way. So the right-hand twist, because it's bDNA. Then if you take that and then you start twisting it back around on itself to the left, that's negative. If you're to take it, that right-hand twist, remember this is my right hand, that right twist, and then you were to take that and start twisting it in a bigger right-hand coil, that's a positive. This positive supercoiling actually helps some of the archae be able to withstand high temperatures, normal high temperatures that would cause the chromosome to unwind, to come apart, to separate. But because of that positive supercoiling beyond just the right-hand twist, it's more stable. Central dogma, as I said a couple of slides ago. DNA to RNA, RNA to protein. Gene expression, that term gene expression, that expression means you're reading DNA, the information in DNA, the blueprint, and you're making RNA. You're using the RNA, the different types of RNA, messenger RNA, tRNA, ribosomal RNA, to make a polypeptide. Now, as I said a couple of times already in this section's lecture, replication, transcription, translation. This is the normal flow, what they call information flow. DNA is the template to make more DNA. DNA is then the template to make RNA. Ribosomal, tRNA, messenger RNA. Those three types of RNA come together, so you get translation, a polypeptide made. So when you look at it graphically, here's a figure. Different DNA polymerases will read the DNA template, make copies. Remember that whole semi-conservative replication that Watson and Crick won the Nobel Prize for. An RNA polymerase that reads the DNA template, what they refer to as the coding strand, and will make a complementary strand of RNA. That complementary strand of RNA may be messenger RNA. The messenger RNA that a ribosome or ribosomal RNA will move down codon by codon so that the tRNA can bring in the correct amino acid. So you have the formation of a polypeptide. There is some differences. The overall scheme of what I've just talked about the past few slides, whether it's eukaryotes, prokaryotes, is relatively the same. It's when you start getting into specifics that there's going to be discrepancies. There's going to be evolutionary divergence. Replication, transcription, translation, the same for all living cells. It's who the players are, what initiates it, what terminates each step. That's going to be different. Another thing is because prokaryotes don't have a nuclear envelope, as soon as that messenger RNA is being made, as soon as it's long enough, It's come out of the, you've got the RNA polymerase marching along, making the messenger RNA copy. As soon as it comes out and it's long enough, boom, a ribosome is going to hop on it. So you can have an RNA polymerase and a ribosome both working on the same messenger RNA. One's making it, the other's reading it at the same time. We know this because we've actually taken pictures of it. Here you look at this, I forget which bacterial cell. This is, it's E. coli. They use different fluorescent markers. The red marker, stained RNA polymerases. The green marker, stained ribosomes. These are the same cells. They just switched the light source. Turned on one light source, everything glows red. Turn on a different light source in the same microscope on the same slide in the same place. Everything glowed green. When they used a computer to overlay the two pictures, hey, wherever there's RNA polymerase, turns out there's also ribosomes, as you see here. RNA polymerase is marching along, making the messenger RNA. Right there next to it is a ribosome reading that messenger RNA as it's being made.